Okay, hi everyone. This is lecture 23, Reproductive. So today we are going to talk about the basics of the reproductive system. We're going to cover the definition of a gamete and how gametes are formed by the process of meiosis. We're also going to touch on sex differentiation in the embryo, that is, production of a male versus a female. And then we will talk in detail about the male reproductive system, the process of producing male gametes, and the female reproductive system, the process of producing female gametes. So I want to remind you of the anatomy for both the male and the female reproductive system, but we're going to start with the male. So the male reproductive system is composed of gonads. The gonads are the most important organ of the reproductive system. So that is where the gametes are formed. So in males, the gonads are testes. And then from there, the gametes, which in males are the sperm, will travel through the reproductive tract. The reproductive tract for the male is the epididymis, to the ductus or the vas deferens, to the urethra, and then through the external genitalia. The external genitalia for the male then is the penis, which contains the erectile tissues, the corpora cavernosa, and the corpus spongiosum. There are also accessory glands in the male that help to support the sperm and to add fluid to the final as the sperm are traveling out of the body. So we have the prostate, the seminal vesicles, and the bulbourethral glands. So the function of the reproductive system is the production of sperm in males, which is spermatogenesis, and the delivery of the sperm to the female. So kind of while we're on this subject, I want to let you guys know that when we talk about reproductive, we are not going to spend a lot of time talking about the actual act of sperm delivery and sperm receipt in males and females. So in other words, we're not going to spend a lot of time talking about copulation. So that is more appropriately covered in a health course that covers the psychology of sex, the actual act of copulation, all of that. We are going to focus on the biology side, which is the production of the gametes genetically and how those gametes are kept alive, how those gametes are induced to mature by the hormones of the male and female reproductive system. So we're going to focus mainly on the gonads and the production of the gametes. So I'm leaving this here for your review in case your anatomy has been a while, so that you can see just in cross-section the male reproductive system. So here are just the important pieces. Here's a single testis in cross-section, and then from there the sperm will travel up through the epididymis, up the vas deferens, which curves around the back of the bladder, then through the ejaculatory duct, through to the urethra, where we have the prostatic, membranous, and spongy urethra, and then out the body. For the female reproductive system, we also have the same organization: gonads, reproductive tract, external genitalia, and accessory glands. So the gonads for the females, which are producing the gametes, or eggs, are the ovaries. The ovaries then produce the eggs, and they will be released every month through the reproductive tract. They will be caught by the uterine or fallopian tubes, enter the uterus, and then if no fertilization occurs and no baby is grown, right, then the process every month will be shed, and the uterine lining will be shed out of the vagina. So the reverse is also true for fertilization. So during the process of fertilization, sperm will enter the vagina, travel up the uterus, and up the uterine tubes, and wait for an egg to be fertilized. Then the external genitalia for the female is not the vagina. That is a common misconception. The external genitalia is the vulva. So the vulva includes several structures, which are the erectile tissue, the clitoris, the vaginal opening, and the labia minora, labia majora. Then we have accessory glands in the female, which are the vestibular glands. So here is a cross-section of the female reproductive system in sagittal section. So here you can see from the outside the vulva structures, the opening to the vagina, and then the cervix, which is the neck portion of the uterus. So then here is the uterus. And then from the uterus, the uterine tubes, which will catch the eggs from the ovaries. So here is an ovary, again traveling backwards through the uterine tube, into the uterus, out the cervix, out of the vagina. And I want you just to notice in cross-section here that the bladder in the female is in front of the vagina, just below and in front of the uterus, and then here in the back is the rectum. So the functions of the female reproductive system are also production of gametes, but the female gametes are called ova, or eggs, and this is the process of oogenesis. Another function of the female reproductive system is also the receipt of the sperm from the male for fertilization, the transport of the ova to meet the sperm if there is any present, and then in addition, for the female, we also have the added functions of pregnancy and childbirth. I wish we had time in this class to talk about pregnancy and childbirth in more detail. There are chapters in your textbook if you guys are curious, but we just don't have the time to get into it. So let's start then and continue our focus on producing gametes. So the gametes are haploid reproductive cells. Do you remember what haploid means? Think back to your DNA lectures. What does it mean for a cell to be haploid? It means that it has only half of the normal complement of chromosomes. So in other words, humans normally have two copies or 2N of each chromosome. That would be a diploid cell. A gamete has half that. It only has a single copy of each type of chromosome. So haploid gametes are 1N cells, and our N in humans is 23. So they are one copy of each of the 23 chromosomes, or 23 total chromosomes in a gamete. The male gamete is sperm or spermatozoa. The female gamete are ova or eggs. Gametes are produced by the process of meiosis. What is special about meiosis is that it creates genetic variability, such that gametes are genetically unique from the cells that they were made from. So this can give the opportunity in humans and other animals that have sexual reproduction for genetically unique offspring. So we will then have a haploid sperm with 23 chromosomes, a haploid egg with 23 chromosomes. Here, this textbook only draws 3 chromosomes, but you get the idea. And then we'll combine them to make a diploid zygote, where half of the chromosomes will come from the mother and half of the chromosomes will come from the father to make a full two copies of each chromosome in the developing zygote. So remember that humans have 23 pairs of chromosomes in a normal body cell. So 23 types of chromosomes and 2 pairs each. That means in every normal body cell or somatic cells, there are 46 chromosomes in human cells. And here's a picture of those chromosomes after they have been pulled out and stained. You can see that each chromosome has sort of a unique morphology to it. And then we also have the sex chromosomes X and Y. So we number them 1 through 22, X and Y. For the gametes, we still have 23 types of chromosomes, but we only have one copy of each. So you'll notice here, this is a somatic cell where you have two copies of each type. So here's chromosome 1 with 2 copies. Here's chromosome 2 with 2 copies. So this is a diploid representation. Gametes, however, are haploid and only have one copy of each type. So haploid gametes are produced in males and females through the process of meiosis. Gametes will then fuse during fertilization to form a zygote. This forms the sexual life cycle in humans. So female germline cells, or the cells that are the reproductive cells, will create through the process of meiosis haploid eggs. Male germline cells or male reproductive cells through the process of meiosis will create haploid sperm. They will meet if there is fertilization, and that will grow as a diploid developing zygote, which eventually grows into a baby. So meiosis is a specialized form of cell division that begins with a reproductive stem cell, which is a diploid cell, 2n equals 46, just like all other body cells. And then through two separate nuclear divisions, meiosis 1 and meiosis 2, haploid cells are produced. So you need to remember a little bit back to the process of mitosis, where you begin with DNA replication, and then you have the phases. So you guys remember this? This is what I did with my hands, right? So we do prophase, metaphase, anaphase, telophase, and then cytokinesis. That was mitosis. Meiosis is going to be one round of a process that looks almost exactly like mitosis with a few very important ways to create genetic variability, and then another complete round of cell division without DNA replication before to simply split from diploid to haploid cells. So meiosis 2 will be another set of prophase, metaphase, anaphase, telophase, and cytokinesis, so that in the end you get 4 daughter cells. I have put here just a quick overview of meiosis 1 and meiosis 2, but I also want to provide a diagram for you guys, so let me show you what I've drawn. Okay, I'm not going to take the time to draw this out in every tiny detail because that would take forever, but I want you guys to take a look at this diagram and I'll take you through it. So here is regular mitosis, and I'm just showing 2 types of chromosomes, chromosome 1 and chromosome 2. Mitosis happens in diploid cells. So we start out with a parent cell that's shown here that has 2 copies of each type of chromosome. I've only drawn 2 types. Maternal homologs in purple, paternal homologs in blue. That parent cell then undergoes DNA replication before mitosis begins, and then we get these duplicated chromosomes in each cell. Then through prophase, those chromosomes condense, line up, separate, and very, very carefully, the way that they are separated is so that you end up with exactly the identical daughter cells from the parent cell. So you will have 2 copies of each type of chromosome and the identical copies that you had in the parent cell. So here's the parent cell, and here are the 2 identically copied cells through the process of mitosis. Meiosis 1 is very similar, except that there are 2 very important differences. So you'll start with a diploid body cell. That will replicate, and then the chromosomes will condense and split. But I want you to notice 2 things about meiosis 1. First, in prophase 1, you have crossover. What that means is that the chromosomes actually will touch and swap genetic material. When they do that, they will now be genetically variable. such that some chromosomes from mom and some chromosomes from dad will swap over, and we will now have genetic differences in the final cells. The second important difference is during metaphase 1, and that is mom chromosomes and dad copies of chromosomes will assort independently. So in metaphase of mitosis, they were very carefully sorted such that you had one mom and one dad copy on either side. In meiosis 1, that doesn't matter. So you can actually completely independently assort, so you end up with mom copies and dad copies on different sides of the cell, and you can then sort that. So even at the end of just meiosis 1, you can have mismatched mom and dad copies of the chromosomes. So here, the way I've drawn it, the purple is a mom copy of chromosome 1, the blue is a dad copy of chromosome 1. They cross over and swap genes, and then they assort completely independently of maternal and paternal origin, such that they can be very different in meiosis 1. And then we will take these same cells from meiosis 1 and divide them up and separate with an extra division without replication beforehand. That will end up with 1, 2, 3, 4 daughter cells now, because we started with 2. And because of that extra round of division and separating chromosomes again, we now only have one copy of each type of chromosome. So the gametes that result from meiosis have half the amount of chromosomes as the parent cell, and as I've kind of tried to draw here with these different dots, those chromosomes will look different because of crossover. They will also be independently assorted, such that you may not always have one mom copy and one dad copy as you do here in the process of mitosis, but you could end up with a bunch of dad copies of chromosome 1 and chromosome 2, or a bunch of mom copies of chromosome 1 and chromosome 2. You should still have the same 23 chromosomes though, they just maternal and paternal origin can be swapped and mixed. So this is why you might look a lot like your grandmother and your sister might look a lot like your grandfather, for example. So meiosis 1 starts with a parent cell. Chromosomes replicate. So here we had a single mom copy and a single dad copy of chromosome 1. They will replicate, and then through the process of meiosis 1, become genetically different through crossover and assort completely independent of whether they were maternal or paternal. And then through the process of meiosis 2, we will get half the number of chromosomes and the final 4 genetically different haploid gametes. So prophase 1 has crossover, metaphase 1 has independent assortment, and this will ultimately result in gametes with a different combination of paternal and maternal chromosomes and mixed paternal-maternal chromosomes. So this mixing here is why you might have your dad's eyes and the rest of you looks like your mom, or you might have your mom's general body but your dad's nose, etc. Okay? So we have crossover that creates genetic variability through meiosis. Here's a picture of what crossover looks like. So we'll have the same chromosome here. This book has also drawn maternal and paternal homologs in different colors. So this is chromosome type 1. This is the one you inherit from your mom. This is the one you inherit from your dad, and they will swap genes. So this is all mom but with dad's nose. And this is all dad but with mom's nose, right? Then they will assort independently. So some moms will go to one side, some moms will go to the other side. It doesn't matter now if we have an equal copy of maternal and paternal homologs. Then meiosis 2 is reduction division. So taking these still duplicated chromosomes and separating them such that each cell is now haploid. These figures I'm going to leave for you if you need a little more detail to help you understand meiosis 1 and meiosis 2. So why do we care about genetic variability? We care about genetic variability because this introduces the potential for so many different kinds of gametes. Genetic variability after sexual reproduction allows new combinations of genes and offspring that have never before existed. This because of crossover and independent assortment during meiosis, but also fertilization between two genetically different individuals creates even more possibility for genetic differences. So humans have 23 pairs of chromosomes. If you do the math, that's 2 to the 23rd possible gametes just after meiosis because of all the crossover and independent assortment that can occur. That's over 8 million possible gametes. Then 2 individuals will combine gametes during sexual reproduction, and that can create 2 to the 23rd times 2 to the 23rd, over 70 trillion possible zygotes after fertilization. So given this math, it's actually amazing that we look like our siblings at all. So now let's get to sex determination. So sex determination is a fascinating topic that we are just going to skim over, but this is whether an individual matures into a male or a female. So this is how anatomically males get male reproductive tract and external genitalia, and females retain female reproductive tract or female external genitalia. There are several factors that go into sex determination. It's not as simple as you might think. So for this, we need to know that, again, you have 22 autosomal chromosomes and 2 sex chromosomes. Males have an X and a Y chromosome. Females have 2 X chromosomes, and this over here, if you look at this individual, chromosomes number 1 through 22 are the autosomal, and then this individual has an X and a Y. So this individual is male. Now, males can contribute an X or a Y then during fertilization. So the sex chromosomes will be split during meiosis along with the autosomal chromosome. So some sperm for males will carry an X and some sperm for males will carry a Y. Their haploid gametes then can have X or Y to donate through the process of fertilization. So it is the males that determine whether a baby from the process of fertilization will be male or female. Females always give an X chromosome. So if you're male, your X chromosome came from your mother. It had to. That's all she could give. And your Y chromosome, if you're a male, came from your father. For females, you have 1 X from your mother and 1 X from your father. So genetic sex determination is determined by males. Who's that guy? Is it Henry VIII that kept beheading his spouses because he was not getting any male children? I think I heard something like that. My history knowledge is terrible. But I think that was it, right? So he kept getting rid of the women in his life because he wasn't getting any male children. Well, it turns out totally his fault. So he was the one, and it is the male that determines whether the baby is going to have an X or a Y in complement to the female's X chromosome. So the first way to determine male or female is genetic sex determination. So this is when an ovum or an egg, which has X or X, combines with a sperm, which is X or Y. So if the mother's X combines with a Y, then you will have a genetically male embryo. If the mother's X combines with a sperm X, then you have an XX, which is a genetically female embryo. So the first piece of sex determination is whether you have an X and an X or an X and a Y. If you are an X from your mom and a Y from your dad, then you are genetically male, X and Y. If you have an X from your mom and an X from your dad, then you are genetically female, an X and an X. But there's more than just genetic sex determination. So the XY or the XX is just the first step. So that's genetic sex. Whether you have an XY or an XX determines male genetics or female genetics. But we also have gonadal sex, and that is whether the gonads actually develop. And that depends on several cues that start with genetic sex, but are also determined by other genetic differentiation. So if you have male gonads, testes, or female gonads, ovaries, then you will also have gonadal sex determination. Then it doesn't stop there. So just having testes or just having ovaries does not make you male or female. Those testes and ovaries actually have to function properly. So they actually have to produce the proper set of hormones, which creates the phenotype of external genitalia. So we have the genetics, which is XY or XX. We have the gonads, which is testes or ovaries. And then if the testes and ovaries are functioning, then we will actually have the external genitalia, which is sort of what we judge each other by, right? So this is penis or vulva. And this is whether an individual has male or female external genitalia. That is phenotypic sex determination. So we already did the XX and the XY. So let's look at the next step. So all embryos actually start out with the same reproductive tissue. No matter what their genetic sex is, they start out with undifferentiated or unspecialized reproductive tissue, which looks the same in an early male embryo and an early female embryo. They all have the potential to develop testes or ovaries, and they're just waiting for the right signal. So on the Y chromosome, there is a gene called the SRY gene. This is a single gene that determines whether testes or ovaries develop. And at about 7 weeks of gestation, or 7 weeks post-fertilization in the embryo, the SRY gene starts to produce a factor called testes determining factor. And it is what it sounds like. If the SRY gene, which is on the Y chromosome in genetic males, produces testes determining factor, then that will trigger testes formation. So you have to be XY. The Y chromosome has to have a functioning SRY gene, and that SRY gene has to produce a functional TDF, or testes determining protein. If all 3 of those things happen, then testes will form. So you got to be XY, the Y chromosome has to have a functioning SRY gene, and the SRY gene has to be appropriately expressed to produce TDF, which will then cause, in the 7-week-old embryo, testes to start developing. If none of that is true, if it is a female or XX genetic female, there's no Y chromosome, so there's no SRY gene and no TDF produced, undifferentiated gonads will simply develop into ovaries. So if there are no signals, ovaries will develop. What's interesting about that is if something goes wrong with the SRY and the TDF, what the developing embryo sees is, no signals, we're going to develop into a female, ovaries. After that, we have phenotypic sex, and that is the actual production of the reproductive tract after the testes or ovaries are formed. So there are 2 reproductive tracts that are present in all embryos. So they will be there from the beginning. The male reproductive tracts in development are called Wolffian ducts, and the female reproductive tracts are called Mullerian ducts. And then one of them has to be selected to actually obtain the reproductive tract. So the selection of the duct depends on testosterone and something called Mullerian inhibiting factor. Both of these will be produced from functioning testes in the embryo. I'm not saying functioning in the terms of they're producing sperm. I'm saying functioning in the sense that they are actually producing testosterone. So they have to produce testosterone and they have to produce Mullerian inhibiting factor in order for the Wolffian ducts, which are now drawn here in blue, to develop into the male reproductive tract. So that is, the testes will form with the SRY gene and TDF, and then the male reproductive tract for the epididymis, the vas deferens, etc., will form with testosterone and Mullerian inhibiting factor. If those signals are absent, then the default is for the Mullerian ducts to stay and produce female reproductive tract. So no signals from the testes or lack of signals because they are ovaries will cause Mullerian ducts to form. So we all start with undifferentiated gonads, the Wolffian ducts, and we all start with undifferentiated gonads and undifferentiated reproductive tracts. The gonads will form based on whether SRY is present, and that will make testes. If SRY is not present, that will make ovaries. The reproductive tracts are both there, and one of them will win based on whether the gonads are functioning. So if the testes are functioning properly and producing testosterone and Mullerian inhibiting factor, then the Wolffian ducts will win, and the female Mullerian ducts will degrade. If there is no Mullerian inhibiting factor and no testosterone, then the Mullerian ducts will be maintained and the Wolffian ducts will degenerate. So then the last bit is the external genitalia. So as with the gonads, the external genitalia are also undifferentiated in early males and females. Then their differentiation depends on the presence of androgens, especially testosterone and dihydrotestosterone, DHT, produced by the testes. So again, we need to have functioning testes to get male external genitalia. The androgens present then will produce male genitalia in this form. So DHT will signal for penis and scrotum development. Testosterone will signal for the reproductive tract development. That is the Wolffian tract I just showed you in the previous page. If there are no androgens or the testes are not appropriately producing androgens, then female genitalia will develop. And here everything is color-coded as to its developmental origin, so you can see the tissues that they originally came from. So this is the undifferentiated genitalia, which starts out with these general structures, and if testosterone and dihydrotestosterone are present, then the male external genitalia, namely the penis and scrotum, will develop. If there is no testosterone and no dihydrotestosterone, then the female external genitalia will be maintained and differentiated. So you can probably hear from some of the examples I have already drawn for you that there are errors that can happen in sex differentiation. So for example, testes can fail to differentiate in the embryo. This is often because there is not enough testosterone. So not enough testosterone is feminizing. That is, a failure to produce enough testosterone can produce a female phenotype. In some cases, this will be a genetic male, so he has an XY chromosome, but is sterile, the testes are not functioning. And if the testes are not properly functioning, then he will be anatomically female. Because the testes were not there, or the testosterone was not there to induce the anatomy of the male to be produced. Lack of testosterone receptors in target cells is also possible, and that is feminizing as well. In this case, the testes are functioning fine, testosterone is being produced, that is genetically male, but anatomically female because the anatomy could not respond to the signals that were being sent out. You can also have a genetic androgen deficiency. This is also feminizing. If you don't have enough DHT for genetic reasons, that is the one that produces the male external genitalia, then internally this individual will look male. This individual will have testes that never descend, reproductive tract in the male that may also never descend, and then female external genitalia, but be genetically male. So in all three of these cases, this person is genetically male but appears female. And that is absolutely biologically possible for many different reasons. We can also have excessive androgen production in females, and that will be masculinizing. So in this case, for example, excess DHEA from the adrenals can take a genetic female during embryonic development and send signals to the genitalia or the external genitalia, and she will look anatomically male. So I think one of the most fascinating things about this is sort of the lack of belief of individuals in the LGBT community when they say, I look female, but I feel male. Or I look male, but I feel female. And I think we have to be careful about judging an individual who says that, because it is absolutely biologically possible to have a mixed male and female type phenotype, or to have a body that looks one way, but genetics that are a different way. So here's a summary diagram, and I think we should just quickly draw this out. Okay, so we have genetic determination, and that is for a male, if you have an XY chromosome, then you are genetically male. But it doesn't stop there. From there, the Y chromosome has an SRY gene. So this is one place where this can go wrong. If for some reason the Y chromosome SRY gene is not there, or it's mutated, or it's not functioning properly, then the SRY gene has to produce testes determining factor, or TDF. That's another place it can go wrong, so it may not actually be functionally producing testes determining factor. If that works, then testes determining factor will produce testes. And that's gonadally male. Genetically male, gonadally male. So he has the XY genetics and the testes present. Then the testes actually have to function and produce testosterone, and Mullerian inhibiting factor. In order for the testes to be appropriately signaling to the reproductive tract. So Testosterone will lead to male reproductive tract, that is epididymis, etc. Müllerian inhibiting factor is to block or prevent the production of female reproductive tract. And in addition to the testosterone, we also from that need to make functional DHT or dihydrotestosterone, which will give external, male external genitalia, and that is penis, etc. So at any point in these processes, you can have issues that cause a genetic male to look female. So you could block the function of the gene, you can block the production of TDF. If the testes aren't appropriately working, then you're not making enough testosterone. If you're not getting enough DHT, etc. So we could do the same thing for females, but I'm going to let you guys go through this diagram and draw out the same thing for females. So genetic sex is XY versus XX. Gonadal sex is testes versus ovaries. And then phenotypic sex is reproductive tract and external genitalia. All right, now that we have developed a male or a female, let's get into then the function of actually making gametes. So for males, gamete production is spermatogenesis, and then we have the process of gamete delivery, which is the process of erection and ejaculation. And we're going to go through both of those pathways for males. So first, spermatogenesis. So spermatogenesis is the process of producing male gametes, or spermatozoa, from undifferentiated germ cells. Those undifferentiated germ cells are called spermatogonia. So I want you guys to go through these stages of spermatogenesis and memorize the names of the different cell types and think about where this is actually occurring in the male reproductive system. So this is in the seminiferous tubules. These are tubules that are located inside the male gonad or the testes. If we take one of those tubules and kind of pull it out and look at it in cross-section, that is what the remaining diagrams are going to show you. For males, this process from undifferentiated spermatogonia to final functional male gamete or spermatozoa takes from start to finish 64 days. But every day since puberty, a male has cells in different stages. So 64 days post-puberty and every day after that, a male produces about 100 million sperm per day that will be maturing into spermatozoa. There are 3 stages of this production: mitotic proliferation, meiosis, and packaging. So this starts in the outermost layer of the seminiferous tubules with the spermatogonia, and then as the cells develop and become more mature, they move to the inner lumen of the seminiferous tubules, where they will then be released into the reproductive tract for the male. So first, mitotic proliferation. This is continuous mitosis of diploid spermatogonia in the outermost layer of the seminiferous tubules. These are spermatogonia, which differentiate into primary spermatocytes. At the end of this mitosis, we will have a new round of DNA replication. So this is just increasing sperm cell number. We're going from spermatogonia to primary spermatocytes. Then we have meiosis. We'll have 2 complete rounds of division to produce haploid spermatids. So we're going to go from primary spermatocytes to secondary spermatocytes to spermatids. But they're not fully functioning sperm yet. They then need to be packaged to fully produce spermatozoa. So this is where the sperm become these tiny little swimming structures that we're all familiar with seeing. So the cytosol and the organelles will be removed, except for the DNA. So within the head of the sperm will be the concentrated chromosomes that will be ready for fertilization. They also have a midpiece and a tail, which helps the sperm become mobile, and they are then mature spermatozoa. So here is where this takes place. This is in the seminiferous tubules of the testes, and now we're going to zoom in here and look at an actual microscope picture of what a cross-section of a seminiferous tubule looks like. And you'll see in the outer layers of the seminiferous tubules, you can actually see the mitosis going on. So you can see the condensing of chromosomes and the mitotic proliferation in the outer layers. And then as the cells move more and more towards the lumen, you'll see once they're in the lumen, these are all the little tails sticking out of the mature sperm that are going to enter the lumen and go out the reproductive tract. So here is a book diagram which shows this process. So remember, this is the outer edge of the seminiferous tubule, and then they will move in towards the lumen as they're becoming more mature. So we go from spermatogonia to primary spermatocytes to secondary spermatocytes to spermatids. That process is mitosis and then meiosis. So you'll notice mitosis is your germ cell. This is kind of like the parent cell that we drew out. And then from mitosis to meiosis, you have twice the number of those cells. And then from meiosis 1 to meiosis 2, you will then have 4 haploid daughter sperm cells. From those spermatids, we now need to make the fully functional and swimming mature sperm. So the spermatids will then undergo packaging to obtain a head, a midpiece, and a tail to become a mature spermatozoa. Males are so easy, guys. Wait till we get to females. So here is the process of spermatogenesis as your book diagrams it, but let's draw it out for ourselves. Okay, so spermatogenesis is going to be in seminiferous tubules of the testes. And we're going to go from the diploid spermatogonia undergo mitosis and mitotic proliferation. From there, the spermatogonia are going to be called primary spermatocytes, and they will still be diploid. From the primary spermatocytes, then, we're going to go through meiosis 1 to form from primary to secondary spermatocytes. So these will now be genetically variable because of crossover and independent assortment. during meiosis 1. The secondary spermatocytes then will undergo meiosis 2 to create spermatids. Now they're haploid. They have the individual single strands of chromosomes, and there will be 4 times as many spermatids as there were primary spermatocytes. Now, one sort of lingering question that students always get confused on is, what are the secondary spermatocytes? Are they diploid or are they haploid? They are actually technically haploid. Now, don't get in the weeds on this one. The reason that they're haploid is because the chromosomes that they have are identical copies, so there is no new information. So we can no longer say that there is a maternal and paternal homologue. We simply have duplicated sister chromatids, and those sister chromatids are not enough to call them diploid. So we split the sister chromatids to make them fully what you guys are picturing as haploid to make spermatids. Anybody needs help with this, please let me know. We can draw it out on the board in class. From there, then, the spermatids will become spermatozoa through packaging. And this is the specialized process of making the head, the midpiece, and the tail of what we commonly call sperm. So that is spermatogenesis. And here's the mature spermatozoan structure that is produced from spermatogenesis. Mature sperm are motile, and they're structured to be able to travel to and fuse with the ova. So this tail is a flagellum, which spins and turns and flips around and makes them able to swim up the female reproductive tract. They will be full of microtubules for that movement to occur, and the midpiece will be full of mitochondria to support that movement with ATP. The head then is basically just the nucleus of the cell, which is a chromosome delivery device. It is covered in something called an acrosome, which has enzymes that are filled to drill into an ovum if an ovum is found. There are some other cells that are nearby that help to support the developing sperm. These are called Sertoli cells, and they're found within the seminiferous tubules, and they provide a lot for the developing sperm. They provide nutrients, they provide protection, they have phagocytic activity, they provide fluid, which will help to flush the sperm into the lumen once they're mature. They also make a blood-testes barrier. We can make a lot of jokes here about a blood-brain barrier versus a blood-testes barrier, but it's actually very similar. And this is preventing pathogen entry into the testes and preventing immune attack of the testes by antibodies. Why would you have an immune attack? Well, because these cells are genetically different from the normal body cells. They could be attacked and recognized as genetically different if the blood-testes barrier were not there. Sertoli cells also provide hormone support. They have something called androgen binding protein, which concentrates and holds on to testosterone to support the developing sperm. They also contain receptors, which will get the signals from the anterior pituitary. They also secrete inhibin. This is negative feedback to regulate the hormone secretion. And then during development, we just learned about this. This is where Mullerian inhibiting factor comes from to make sure that the male reproductive tract is maintained and the female reproductive tract is degraded in a genetically and gonadally functional male. So now let's talk about the hormone control of spermatogenesis. You guys are hormone experts now because we've finished the endocrine system, so this should look really familiar. You're going to have a releasing hormone from the hypothalamus. So the hypothalamus releases GnRH, or gonadotropin releasing hormone. GnRH is stimulating the anterior pituitary to release two tropic hormones, LH, luteinizing hormone, and FSH, follicle stimulating hormone. These are not named for their role in males, they're named for their role in females. So LH will stimulate testosterone secretion through the Leydig cells in the seminiferous tubules. These are cells nearby, not the Sertoli cells, but cells nearby in the seminiferous tubules that are endocrine cells. FSH will stimulate the production of inhibin, which will feed back on the anterior pituitary. Once the Leydig cells produce testosterone, testosterone will increase spermatogenesis and support spermatogenesis. Testosterone is also important for many other basic male sex characteristics and male development. Testosterone will also negatively feed back on the anterior pituitary and on the hypothalamus to keep its levels in check. So LH stimulates the Leydig cells to produce testosterone. FSH stimulates the Sertoli cells to produce inhibin. Both of those will increase spermatogenesis and cause sperm to be matured and produced by the testes. And inhibin will feed back, and testosterone will feed back on the system to keep everything in balance. So this is the last bit that we'll talk about for males, and then we're going to move on to females. So once you make the sperm, you then have to deliver the sperm. And this is achieved by two spinal reflexes. And I think it's only fair because males have to sit through the menstrual portion of the reproductive lecture that females have to sit through the erection and ejaculation portion of the lecture. But in all seriousness, whether you are male or female, I think that it is very important as healthcare practitioners that we take the reproductive system very seriously and that we understand for our patients when the basic reflexes can go wrong, because reproductive health is a very important part of our overall health. So for males, the two spinal reflexes are erection, which is engorgement of the penis, and this actually happens with increased blood flow. How do you get increased blood flow locally? Vasodilation. Vasodilation, we've already talked about in the cardiovascular system, can be increased by a signal called nitric oxide. So nitric oxide will be induced by the parasympathetic nervous system. This will cause local vasodilation in the arterioles of the penis, and it will cause the erectile tissues, the corpora cavernosa and the corpus spongiosum, to engorge with blood. It will also cause secretion of mucus from the bulbourethral glands, and then ejaculation will be the actual release of semen. So, interestingly enough, this is a sympathetic reflex. So normally we think of parasympathetic as the rest and digest and supporting sort of the basic functions, including reproduction. But here's an exception to that. The sympathetic nervous system is responsible for the ejaculation reflex. And this is contraction of the prostate, contraction of the ducts, and contraction of the seminal vesicles to move fluid into the urethra. And this fluid will carry the sperm into the urethra so it can exit out of the male reproductive tract. The bladder sphincter will close because the urethra is a shared passageway for urine and semen, and it will prevent entry of sperm into the bladder. It will also prevent entry of urine into the urethra at this time, so that only semen can be released out of the male urethra during the ejaculation reflex. Contraction of skeletal muscle then will occur at the base of the penis to release the semen. And that's all we're going to say. We're going to move on to the female reproductive system. So the female reproductive system is the production of female gametes, which is oogenesis, and then delivery of the female gametes, ovarian and menstrual cycles, and unfortunately we don't have time to get into pregnancy and childbirth. So oogenesis is the first one that I want you guys to really pay attention to and take careful notes, and be careful step by step to look at each of these phases before you move on to the next. So go slowly here, guys. I'm presenting it in this order for a particular reason. So oogenesis is the process of forming female gametes. These are ova from germ cells. So the germ cells are called oogonia, and they are located in the ovaries. This begins prior to birth and is arrested in various stages until hormonal cues trigger maturation of 1 ova per month. So the stages of oogenesis are complex, and it starts with mitotic proliferation. It goes through meiosis, but meiosis is unusual in oocytes. It gets stopped at various phases during development. It's going to stop before birth and stop during the monthly cycle. So I'm going to divide those up into meiosis part 1, part 2, ovulation, and fertilization. We're going to draw this as we talk about it because I think it's going to make more sense if we draw it a bit. So oogenesis is going to happen in the ovaries, and it actually starts before birth. This is a very unusual difference between males and females. So we start with oogonia in the embryo, and the oogonia undergo mitotic proliferation by about 5 months in the embryo. From there, they will turn into primary oocytes. Primary oocytes are going to undergo partial meiosis 1 up to metaphase 1. So up to metaphase 1 means that they have undergone crossover and independent assortment. So they are genetically variable. There's about 2 million of those at birth. So at birth, a woman has all of the primary oocytes that she will have for the rest of her life. From there, the primary oocytes arrested in metaphase 1 are going to go through meiosis 2, are going to continue to complete meiosis 1 at puberty. But not all of them do. So we have about 2 million primary oocytes that females are born with, and then at puberty, only 1 or 2 out of that 2 million will be selected by hormone signals to undergo and complete meiosis 1. As that happens, it becomes a secondary oocyte. And it's going to stay a secondary oocyte throughout the monthly cycle, and it will only complete and become a mature ovum only if fertilization happens. Otherwise, the secondary oocyte is that which gets ovulated and travels through the female reproductive tract for fertilization. Only if fertilization happens does a mature ovum get finally produced, and a mature ovum is finally produced by the completion of meiosis II. So some oocytes will never get to complete meiosis II. Actually, most oocytes will never get to complete meiosis II. Most of them are going to be these secondary oocytes, which have only completed meiosis I. One last detail here is that when meiosis I is completed, you're going to end up having one cell that hogs all of the cytoplasm and gets to be the secondary oocyte. And the second cell, which will be the second daughter cell of meiosis I, gets degraded and is called a polar body. The same thing will happen with meiosis II. You'll take this secondary oocyte and that will divide by meiosis II. One of them is going to hog all of the cytoplasm, and the second one will degrade into the second polar body. So here's the book diagram of that same process, where you have oogonia undergoing mitotic proliferation and arrested at metaphase I in the primary oocyte at birth. From there, the primary oocyte will become a secondary oocyte during puberty. The secondary oocyte is what will be ovulated and complete meiosis I, but it will not become a mature ovum unless there's fertilization, and that's why a sperm is drawn here, which is signaling to the oocyte to become a mature egg as fertilization occurs. Or differentiation of the egg. We're not done. Female reproductive system is now more complex because the oocytes are supported by a very important structure called a follicle. So if the oocyte is the gamete, the follicle is its house. The follicle supports the cells as they grow and mature. So it is a layer of supportive cells that also change and grow as the oocyte grows. Follicles are made up of cells called thecal cells and granulosa cells. They provide nutrient support and estrogen production to the developing oocyte. And here is a view of what the follicles look like. And I want to go through the follicle types and draw them with the egg as we talk about them. Okay, so we have the oocyte, which started out as an oogonia and then became a primary oocyte at birth. The primary oocyte then is going to wait for the appropriate hormonal signals to mature into a secondary oocyte. That secondary oocyte is what gets carried all the way through ovulation and waits for fertilization. Only if fertilization happens does it become a mature ovum. So the egg spends most of its reproductive cycle as a secondary oocyte during the month of ovulation and impending fertilization. But the egg spends most of its life cycle as a primary oocyte, waiting for the hormonal signals to trigger its monthly cycle of ovulation. Then we have a follicle. In the embryo, we have primordial follicles. Primordial follicle is a tiny cluster of follicles. So a primordial follicle really doesn't have much structure to it. It's very small, and all of these guys will be clustered at sort of the edges of the ovary. Primordial follicle will contain oogonia, and then primary oocytes will be surrounded by primary follicles. So the primary follicle begins to get a little bit bigger. So there's an oocyte, and it will be surrounded by a larger set of follicle cells. So I want you guys to notice that I'm drawing the oocyte as a black dot. So as we go through the oocytes, primary, secondary, mature, they are very tiny cells relative to the follicle. And they are not going to look that much different as we go through. It is the follicles that look different that you will be able to recognize. So the primary follicle contains a primary oocyte, and then the primary follicle, based on hormonal signals, is going to start to increase in layers. So you're going to get more and more layers surrounding the oocyte. And that is now a secondary follicle. So a secondary follicle contains secondary oocytes. And a single group of these, based on hormonal signals, will start to mature further. So there's our oocyte, and as it matures further, it may take on a little bit of fluid, and it's going to get more and more cell layers around it. These we still call secondary follicles until we get enough fluid that we have this huge pocket of fluid called an antrum, which surrounds the oocyte, and then a huge layer of cells now making up the follicle. These follicles are very big compared to primary follicles, and these are the follicles that are eventually going to rupture and open up and release the egg. So during ovulation, these mature follicles, which are also called Graafian follicles, will ovulate, and the egg will get released. So these are all secondary follicles with secondary oocytes until we hit a Graafian follicle, but it's still a secondary oocyte. So these are all containing secondary oocytes, and is the secondary oocyte that leaves and looks for sperm, or waits for fertilization. Only if fertilization happens does it become a mature ovum. but it is no longer in the follicle. It is now in the reproductive tract, and the follicle stays behind in the ovary and becomes something called a corpus luteum. So this is an ovum, this is a secondary oocyte waiting for fertilization, and a corpus luteum that is also waiting for fertilization. Okay. The corpus luteum is an important structure that secretes progesterone. So let's now look through, we've kind of drawn them, but I want to show you now the pretty pictures, right? So we have the primary follicle, which contains a primary oocyte. We have a secondary follicle. There's another secondary follicle in a later stage, which contains a secondary oocyte. We then have mature follicles, which still contain secondary oocytes. That secondary oocyte will be ovulated, given the right signals, and the follicle will stay behind. The follicle will stay behind and become this yellow structure called the corpus luteum. The corpus luteum actively secretes progesterone, and if no fertilization occurs, then the corpus luteum will degenerate into something called the corpus albicans. So luteum is a yellow body, albicans is a white body. So the corpus luteum is nice and yellow and secreting progesterone. The corpus albicans is a degenerating corpus luteum. So we have this summary diagram, which should make sense now that we've talked about every piece of it. And I want you guys just to pause here and take a moment to make sure you can find the oocyte and find the follicles surrounding each stage of each structure. Good? Okay, so now that you recognize the morphology of the follicles, let's talk about the role of the follicles in hormone secretion. So developing primary follicles secrete estrogen. This will stimulate follicle growth and maintenance. Secondary follicles will peak estrogen secretion. So the more the follicle matures, the more estrogen will be secreted until we reach a peak in estrogen. And so if you look down here at this graph, which tracks estrogen production, you can see as the follicle goes from a primary to a secondary, to a late secondary, to a mature Graafian follicle, we ultimately reach a peak of estrogen. So more and more estrogen is produced as the follicle is maturing. After that, ovulation will happen, and ovulation will leave behind the corpus luteum. The corpus luteum now knows that an egg has been sent out into the world to find its mate, right? So the egg is now traveling through the reproductive tract looking for sperm, and the corpus luteum stays behind and secretes progesterone. Progesterone is before pregnancy or supportive of pregnancy. So progesterone is now going to increase as the egg is out there looking for sperm to be fertilized. So the estrogen will peak as the follicles are maturing. The progesterone will peak as the corpus luteum takes over. Progesterone stays up until the corpus luteum degrades, and then after the corpus luteum degrades, progesterone goes back down. So here's a zoom in of that same graph, just so that you guys can see it a bit better. So FSH and LH are also used during the female reproductive cycle. This is again the same hypothalamic pituitary axis of control we saw for many other hormones, but it's a bit more complicated for female reproduction because low levels are different than high levels in terms of how they cause changes. So here is basically how it goes. The hypothalamus secretes the releasing hormone, gonadotropin releasing hormone. That will trigger the anterior pituitary to secrete LH and FSH. Follicle stimulating hormone, or FSH, triggers the follicles to mature because they stimulate follicles. So they are named follicle stimulating hormone because that increases maturation of the follicle. LH also triggers an increase in maturation of the follicle. So both LH and FSH send the signal to one or two follicles to mature. Once the follicle matures enough that it secretes high levels of estrogen, that high level of estrogen will feed back on the anterior pituitary. It's going to feed back on the anterior pituitary and actually stimulate LH. Very high levels of estrogen stimulate LH. So LH knows that the follicle has become mature enough that it is time to ovulate. So the stimulation of LH leads to a huge peak in LH. We call the LH burst, and that triggers ovulation. The egg will now go through the reproductive tract to search for sperm to be fertilized, and the follicle stays behind and becomes the corpus luteum. The corpus luteum will secrete progesterone and estrogen together. So here is that same diagram, just a little bit bigger so you guys can see it. Now that the corpus luteum secretes progesterone, high levels of progesterone and high levels of estrogen are going to feed back and inhibit LH and FSH production to prevent any other follicles from maturing. So an egg is out there looking. The corpus luteum is supporting pregnancy with progesterone. I'll show you how in a minute. And it is also making sure that no other follicles or eggs will be maturing so we can focus on what is already out there. So now let's talk about how the ovarian cycle and the cycle of the uterus work together. So the ovarian cycle is what we just did. That is the state of the follicles and the hormone secretion cycles every month. But we're going to more carefully look at each of those stages. The ovary or the ovarian cycle then will dictate the uterine cycle, which is the lining of the uterus, where if there is fertilization, an embryo would grow. So the uterus will be prepared for a potential pregnancy as the eggs are being released. There are several phases to the uterine cycle as well, and we're going to put it all together at the end. So first, let's look at this diagram just in the follicles to make sure you guys are following so far. So we have the primary oocyte in a primary follicle, the secondary oocyte in a secondary follicle. That follicle will then develop into a mature follicle, and the secondary oocyte will be ovulated. The follicle will then the follicle will then become a corpus luteum, which secretes progesterone. As the follicle matures, estrogen increases and then goes down. As the corpus luteum is produced, progesterone increases, and then will return if no fertilization happens. The high levels of estrogen cause a peak in LH that triggers ovulation. FSH will go up in the beginning as FSH and LH are increasing to increase follicle development. So let's look at the specific timing of these cycles. The ovarian cycle is the state of these follicles and the hormone secretion cycles every month. So we start at day 0, which we call the follicular phase. This is when FSH and LH are rising and stimulating follicle maturation. This will then stimulate oocyte development, and the estrogen will slowly rise and then peak as the follicle is growing. So the follicle itself is producing this estrogen. So as the follicle gets more and more mature, the estrogen will slowly rise and then peak. That peak in estrogen triggers a surge in LH, FSH happens to follow, which is a burst that triggers ovulation. The oocyte will then leave the mature follicle and travel through the reproductive tract. The corpus luteum will stay behind and start the luteal phase. So 0 to 14 is the follicular phase, where you're going from a primary follicle to a secondary and then a mature Graafian follicle. Day 14 to 15 is ovulation, where the Graafian follicle is releasing the egg. And day 15 to 28 is when the corpus luteum takes over. So the corpus luteum will cause high levels of progesterone, moderate levels of estrogen, and it will inhibit LH and FSH. The inhibition of LH and FSH is important for preventing other follicles from maturing while one egg is out there looking to be fertilized. This is also why birth control works. Birth control is usually some combination of progesterone and estrogen that tricks the body into thinking you're in the luteal phase and an egg has already been ovulated. So no further eggs will be stimulated to grow and ovulate. So here are the FSH and LH levels during day 0 to 14. So that is stimulating oocyte development and follicle growth from day 0 to 14 until the follicle grows enough that an LH burst occurs, and then ovulation will happen day 14 to 15. And then after that, the LH and FSH will go way down because they're going to be inhibited by progesterone until another cycle begins. So we can track the development of the follicle, FSH and LH, and estrogen and progesterone based on what is happening in the ovaries. So day 0 to 14, follicular phase, the follicles are developing. Day 14 to 15, ovulation. And day 14 to 28, corpus luteum takes over. All of this also sends signals to the uterus. So the follicles in the ovaries producing hormones will send signals to the uterus that we're in a different stage, and the uterus should prepare for that stage. So day 0 to 7 is actually cleaning house from the previous cycle. We'll get to that in a second. Day 7 to 14 is in preparation for ovulation. So as estrogen is rising and the follicle is maturing, day 7 to 14 of the uterus is where the uterine lining begins to thicken. So this thickens in response to increasing estrogen from the ovaries. By day 14 then, by ovulation, the uterine lining has thickened, and it continues to thicken and even become secretory up until day 28. So as the egg is ovulated and then travels through the fallopian tubes and down into the uterus, the uterine lining will become thicker and thicker and more secretory due to the progesterone that is secreted from the corpus luteum. So progesterone from the corpus luteum is supporting pregnancy primarily by supporting the uterine cycle, and that is thickening the lining of the uterus so that it is ready for the implantation of a potentially fertilized egg. If there is no fertilization, no implantation of the egg into the uterine lining, then it is time to clean house and start over. The uterine lining will be shed, and there will be the menstrual phase as that uterine lining is shed through menstruation. If fertilization occurs, then pregnancy will happen. So day 0 is either the menstrual phase or continued pregnancy. So now we can look at the estrogen increase that is from the follicles maturing, and that causes some increase in thickening of the uterus lining. And now the progesterone increase during the luteal phase causes an even further thickening and secretory state of the uterine lining. So this is making a comfy, cozy, blood-filled secretory uterus that if there is a fertilized egg, it can dig into the lining of the uterus, implant, and have a safe place to grow and develop. This happens specifically in the endometrium of the uterus as a result of first estrogen increasing and then progesterone increasing. And as long as progesterone is high, the uterine lining will be thick. Once progesterone declines, the uterine lining will get the message that no fertilization has occurred, and it will be shed to restart for the next month. Okay, are you guys getting it? Take a moment to go back to the summary diagram, this one, and go step by step. What is happening in the follicle? What is happening in the egg? And what is happening in the uterus that mirrors what should be happening with the egg? Make sure that you understand the estrogen levels, the progesterone levels, and the FSH and LH levels that occur because of that. Make sure that you can track both the follicle stages in the ovary and the uterine stages that support the potential for the fertilized egg to implant into the uterus if everything goes properly. Okay, that's it. That is the end of the final lecture for this semester. Please let me know if you have any questions.